Sorci-Thomas's third visit, I believe, here, but the first time you've actually been around.
Yes.
So if everyone remembers, we invited Dr. Sorci-Thomas to come here for a seminar.
It's true.
I'm sorry, our Nutrition Symposium, and the flight never landed. So this is actually the second time we've had you scheduled here. So
Yes, thank you.
And almost didn't make it this time too, actually. So,
You got stuck in Dallas for a while, but
so a couple notes on Dr. Sorci-Thomas. Dr. Sorci-Thomas received her Bachelor's of Science in Biochemistry at Louisiana State University and her PhD in Biochemistry at Wake Forest University in North Carolina. She also had a prestigious NIH National Research Service Award and completed a postdoc fellowship at the State University of New York. She has chaired over 20 committees regarding lipoprotein metabolism for the American Heart Association. She also has served as an ad hoc reviewer for over 12 panels and committees for the NIH National Heart, Lung, and Blood Institute. She served on the review committee for Pfizer International HDL Awards Program, which granted over $6 million to support basic and clinical research on HDL metabolism. She currently serves on the editorial board for both the Journal of Lipid Research and Arteriosclerosis, Thrombosis, and Vascular Biology. Finally, she served as a peer reviewer for over 10 scientific journals, including Atherosclerosis, the American Journal of Pathology, and Circulation, and has over 65 journal articles published to date. And I counted those. Dr. Sorci-Thomas' research is focused on the mechanisms and chemistry associated with HDL biogenesis and HDL conformational lipid accumulation capacities in order to understand the inflammatory processes associated with chronic disease, in this case, atherosclerosis. She currently holds over 3 active NIH grants dealing with this topic. Dr. Sorci-Thomas' work has been instrumental in defining what we currently understand about HDL and its implications in chronic disease prevention and possibly in some of its associations with causing chronic disease disease. And with that being said, please join me in welcoming Dr. Sorci-Thomas.
Thank you. That's probably one of the best and more accurate introductions that I've had. Thank you. And I was bound and determined to get here after what happened in March. Seriously, we don't have to relive that. But when I got to O'Hare and there was a mechanical problem with my flight, and then that caused me to miss my 4:30 connection to College Station, I called Rose and said, I don't care, I'm coming if I have to get on a bus. I'm not the luckiest person, and I'm also not jinxed, and I was just tired of not being able to get here. So anyway, it is really a pleasure. And being an LSU Baton Rouge grad, it's very nostalgic, if that makes sense. Probably doesn't, but I'm living in Milwaukee now, and trust me, it's anything but like the southern part of the US. So what I want to do today, I'm going to talk, and I apologize, I brought my laptop, and there, might be some issues with slides that don't exactly fit on the screen. But what I want to talk about today is my research program, and I see so many young faces out here that I am hoping we will be more interactive and less, thinking about whatever you did over the weekend or whatever. And so I really want to encourage you to interrupt me during my presentation with questions, because what I'm going to cover is really new, and it's new for me, so I assume it's going to be new for you as well. This is our medical center complex in Milwaukee. Believe it or not, this was not the only day of the year that there wasn't snow, which is what I get a lot. The atrium of the building I work in, and a beautiful picture on Lake Michigan. Let's talk about HDL a little bit. I think this figure may be quite familiar to many of you. It is taken from the Framingham Heart Study. And what we have here are cholesterol groupings of cholesterol values for human beings. And this group of individuals would have LDL cholesterols around 100 milligrams per deciliter. And in this category, very high HDL, okay? And you can see as the LDL increases at a set HDL, your relative risk goes up, okay? So that we all know, right? This is why atorvastatin and lovastatin and all the statins make billions of dollars a year for pharmaceutical companies, because the idea is to lower your bad cholesterol. Now, the other side of the coin for me is HDL cholesterol. So very precious few people have HDL cholesterols that are 85, okay? And probably many of you, if I asked you, you'd probably raise your hand, you know what yours is. Typically, we tend, most of us in the 45 range, but there are individuals at 65. And then the folks that are at 25, you can see even at the lowest LDL, they are still at the highest risk or higher risk for coronary events, okay? So these are data that have been accumulating for many, many years. And anytime people do these type of studies, there are significant correlations. So it has become in the public to understand that you want to have the highest cholesterol, HDL cholesterol as you can. Now, the problem with this is not with the epidemiology or the studies, it's with reality. And the reality is that there have been several attempts at pharmacologically raising HDL levels, which have worked, but the relative risk has not been reduced, okay? And that has caused a rethinking in the area, and that is, what are we really, what is really being measured here, okay? Now, I don't know, do they teach a course in lipids here for students? So everybody knows what cholesterol looks like, kind of, and phospholipid, kind of, okay. Okay.
Oh, wow.
Well, I'm not going to test anybody, but anyway, but I want you to visualize what you may think an HDL looks like. I'm not going to really talk about that, but what I want you to think about for a second is that this is what we typically do. We get plasma from people and we measure things in them, and some of us do it very well, some of us not so well, but in these large studies, they are measuring HDL cholesterol concentration. Now, if you think about that, that is a snapshot, okay? Because really, HDL is synthesized and it's catabolized. And so whatever is in your plasma at any one time, or even several times over a course of a month, what we call that is a steady state. Okay, if it wasn't steady state, then your HDL level would go way up, right? It'd be up in 85, and then eventually you'd become 200 and 600, right? Because you have to have an outlet. As production increases, the outlet has to increase if you're in steady state. So what the field started thinking was, okay, maybe these steady state measurements, maybe they correlate with prevention or reduced risk, but maybe there is a better measure, particularly as we look at individuals and say, okay, are you at risk for coronary disease? We can put this data into, you know, a formula, an algorithm. Actually, the American Heart Association does that, right? Has anybody ever been on the American Heart Association website and looked at their risk calculator? I have. Yeah, it's pretty cool. So you put in like age and some of these measures, you know, body mass index, glucose or whatever. So, so maybe we can make the algorithm more sensitive. That's the idea. Okay, so just a quick review of what I was saying. So the liver makes VLDL and LDL, and the idea is that this stuff, this cholesterol, which is in the core and triglyceride of these particles, gets into the intima of your coronary artery, and that causes you to be susceptible to coronary artery disease. HDL being the good guy and ApoA-I being my favorite protein and one you're going to hear more about than you ever probably have before, is theoretically the good cholesterol, and somehow it magically takes the cholesterol out of the artery that the bad guys deposited. And of course, as I'm sure you all know, being good nutrition students, that the only site of cholesterol catabolism is excretion by the liver, right? You don't catabolize it like you would a fat or a carbohydrate or an amino acid. Okay, so what is now the trending method? The trending method is to look at cholesterol efflux capacity. Now, what does that mean? I'm just showing you a table from a paper that came out from Dan Rader's group in 2011 on the New England Journal of Medicine. And what Dan hypothesized, he and George Rothblatt, that the ability of a person's serum HDL somehow could be used to measure this, I'm going to call efflux, okay? To get cholesterol out of a cell or a tissue, we refer to that as efflux, okay? So then what Dan said was, okay, we can take plasma from patients and we can measure their HDL's inherent property to efflux cholesterol. And so the way they did this, and these are just the results, they got a huge study population, huge for this kind of assay, I would say, and they had healthy volunteers, case control patients with angiographically confirmed coronary disease, okay? And what they found, they calculated the odds ratio, and as you can see here in red, they show that the efflux capacity was a better predictor than HDL cholesterol. And this is then history. So now this study, just this year, was Phil Shaw down the road at UT Southwest in Dallas, collaborated, but did a study with the Dallas Heart patient samples and confirmed this result. Okay, so now here comes the interactive part. What do you...
Texas too. Yay.
Yeah, well, I mean, and it was like LDL country,
Yeah.
right? Yeah. So I thought that was even more spectacular. Okay, question and answer part. So what is it in the HDL that's driving the efflux? Any ideas? I mean, did I give you a hint over here? Oops. ApoA-I, thank you. This is a gimme, come on. All right, but how does it do that? So, I mean, I'm not going to get real heavy duty in the mechanism or anything because I just don't have enough time and I kind of want to get to what our project is. But I wanted to give you a little something that hopefully, you know, as this whole area continues that you'll remember. But basically, this efflux capacity is probably driven by ApoA-I. And the truth is, the way this assay is performed, it's actually quite difficult. You have to take cells in culture. You load them with tritiated cholesterol to equilibrium, and then you add the patient's plasma. Okay, can you imagine 800 wells and doing it in triplicate and all this variation? The point is, if it was just measuring ApoA-I, isn't that just like an ELISA assay? Yeah, so why go to all this trouble to get what is a kinetic term? It's an efflux capacity. So, you know, stay tuned because even the guys that develop this do not have the answer yet, or at least they're not talking about it. Okay, so let's just talk a little bit about chronic inflammation. So I've told you what LDL and HDL potentially do. So why do we get athero? Okay, is it just because we have LDL that we get atherosclerosis? Has anybody here ever worked with an animal model that doesn't have much LDL? Probably wouldn't have known it, right? You might have worked with an animal model but didn't know. So actually a mouse and a rat have very little LDL. I bet you didn't know that. They have mostly HDL. Of course, they don't eat Big Macs either. So it's kind of a genetic environment issue like most things. But what the current dogma is that immune cells in circulation, they become activated with either oxidized or modified LDL in circulation or in the intima, and that this causes this cascade of cytokine production. So as the cells become sick, they give out more cytokines, calling in yet more cells. And so the elevation in LDL cholesterol is actually kind of part of the problem, okay? Now, our way of thinking is that there's actually a component of immune cell signaling that is involved, and we are very interested in lipid rafts and immune cell signaling. So, but that's not really the story I'm going to tell you today. But I just want you to be aware that cholesterol is clearly part of the culprit, because if there was no cholesterol in our diet, your body still makes plenty of cholesterol, and you have to have it. It's an integral part of all your cellular membrane. But the truth is that when it becomes a chronic inflammation, it's when the cytokines are calling in more cells that you get what is shown here as a necrotic plaque. And this, all the connective tissue here can be damaged, and then you can have what they call an unstable plaque. And these are just a few of the cytokines. I just like to show this and see people's eyes roll when they go, oh my God, is she going to really talk about this? No, I'm not. But it's fun to look at. Okay, what I am going to talk about is some of the connective tissue. And we actually weren't even all that interested in this, but I'm going to show you where we got kind of drawn into it. But I want to remind you again of what the disease is that we're studying, okay? So remember, atherosclerosis, it is the #1 reason people die in this country, okay? More people die of cardiovascular disease than all the cancers and everything else combined, okay? So not only is it a huge health problem, but pharmaceutical companies are very keen to make money or save lives, or however you want to look at that. And so what you're talking about is your arteries. I'm glad, some people thought that was funny. And what you're trying to do is you're trying to keep that vessel from accumulating plaque. Okay, so it's kind of like plumbing, really, right? You just don't want deposits on the wall of the pipe. And you've got things floating through your blood that you really can't do without. I mean, as much as people poo-poo LDL, it carries all kind of very essential vitamins and nutrients and so forth, okay? So you need it. We don't need oxidized LDL, but oxidation happens. And so you have this potential. What causes the complication are the immune cells, of course, also you need. And then you have smooth muscle cells that when this cytokine storm is occurring, you have a lot of proliferation. And this causes maybe some narrowing, but also instability. And so where the connective tissue comes in is this keeps that pipe intact. But as necrotic material accumulate, this is broken down. And this is actually what some people believe that aneurysms are caused when these, the collagen and connective tissue become weakened, and you can actually have you know, kind of a release of blood and so forth and die from this. So the idea is that you need to have constant tissue repair if you are going to have constant damage due to cytokines and cholesterol accumulation. So enter in my project. So why did Mary ever even start to think about extracellular matrix or connective tissue? And that's going to be in the next, but I just want to remind you that this would be the cell membrane, okay, or the plasma membrane. And in that membrane, you have integrins, you have collagen fibers, which have proteoglycans like heparan sulfate proteoglycans, and you have fibronectin and laminin, and a lot of things that maybe you guys think about, but I never thought about. And now I have an NIH grant to study this thing, which most people can't stand to try and pronounce. We just call it PCPE2. And I'm going to tell you a little bit about it and then tell you why we're studying it. It's 52 kilodaltons. It is a glycoprotein. It is found in the extracellular matrix, bound to these kind of tree structures, the proteoglycan structures here. And what little is known, there are probably like maybe 6 publications on it or 10. It is known that it assists in pro-collagen processing. I didn't know anything about that, okay? And it's highly expressed in heart, aorta, adipose, and liver being the least. But what really caught our attention was the heart and aorta. And then I was at a meeting. I'm gonna skip some of this history in case anybody is interested. But I was at a meeting, and the senior author on this paper gave a presentation, and he showed that there was in humans there was a correlation between PCPE2 and serum HDL cholesterol concentrations. And they went on to actually find someone who had made PCPE2 global knockout mice, and they had made it for a completely different reason. And what they noticed was that the serum efflux capacity was very reduced in these mice, okay? So remember, we talked about the efflux capacity was a better measure than just looking at HDL cholesterol in terms of predicting like odds ratio for heart disease. So when they tested that, it was actually lower, but get this, the particles, now this is a gel that just shows the size of the HDL in like wild type mice versus knockouts. Look how big that particle is. So this is Dalton's, I'm sorry, these are nanometers, or you can get like a diameter, basically. I mean, it's, my electron microscopy would have been better, but these are what we call non-denaturing gradient gels. And so what got me sucked into this project was the idea of having reduced efflux capacity with an enlarged HDL particle. It was kind of a paradox, right? Because the way we've been thinking in the field is the more HDL, the better, and maybe in some cases the larger. But in mice, you don't have any CETP, which I'm not going to really talk about today. But anyway, so there's obviously a complication of mice to humans, but the two things that sucked me in was this relationship in humans and this paradox. Does anybody want to ask a question? Because I'm just racing on, and if you aren't really comfortable at this point.
Can you explain a little bit the distinction between an HDL concentration versus an HDL cholesterol concentration?
Great question.
The relationship between particle diameter and cholesterol load?
Yes, that's a great question. And I kind of glossed over that because, you know, at some point it's actually very difficult, and another way it's kind of simple. So when you think of HDL cholesterol, The cholesterol is kind of the guts. It's the core of the particle, right? You're talking about a piece of fat that has to circulate in your blood. So how does that work, right? I mean, you've mixed oil and vinegar. They don't mix. So the cholesterol and the triglycerides are the oil. So how do you get that to mix? Because if they don't mix, it just sits there, right? So that's what ApoA-I does. ApoA-I is a detergent. Everybody wash their clothes? You ever wonder how the detergent gets the grease stain out? Well, that's what ApoA-I does. It's a detergent, seriously. And I could give a lecture on what I used to do in another life. But the point is that you can measure the guts of the particle, or you can measure the protein around the particle. As the particle gets more cholesterol, ester, it gets larger. Okay? So pharmaceutical companies, in all their wisdom, have been wanting to raise HDL cholesterol levels, because you saw that Framingham slide that showed the higher the HDL, the less risk, right? But if you make a particle that's the wrong size, to me, the way I explain it is you're looking at a particle that can't be catabolized. It may represent a defect in the pathway, and we're going to get there if you guys will stick with me and ask questions, please. I know there's another question. Come on, somebody, please. She'll give you extra points. No? Just one? Okay, I'll keep going. So what is it? I mean, they're different measures, but they should all correlate. So if you have more HDL cholesterol, you should have more ApoA-I. It doesn't really tell you about particle size. And that's the rub, okay? And that's why we have a lot of sophisticated techniques. Okay, so moving on. So if you look at these mice, which we did, and we just, before we figured we would do a whole lot of work with them, we just wanted to see if we could replicate what was in the literature. That's always a good place to start. And sometimes it doesn't work out the way you want. But anyway, we were able to show that the HDL cholesterol was in fact higher in the knockouts and as compared to control mice. And there was a suggestion of a higher triglyceride, and indeed there was a higher A1. So just like to answer Rose's question, you have an increase in particle cholesterol, HDL cholesterol, A1, and in size. It doesn't always work that way, but in this case, that's how it worked. So that was good. And then we thought, okay, let's cross these animals onto a hypercholesterolemic background. Okay? So the use of the LDL receptor knockout, that down the road in Dallas, the Brown and Goldstein empire, they won the Nobel Prize for discovering the LDL receptor pathway. Okay? I bet everybody in here knew that, right? No, you should know that. They should know that. Now you know it. Okay? Don't forget it. It's pretty cool. Familial hypercholesterolemia is actually quite common in the human population in the heterozygous form. Anyway, what they were able to work out is that these mice look more like humans if you knock out their LDL receptor. And that's all I'm going to say about it. So most people use this as a, quote, control. And what we did was we crossed them with the PCP2 knockout, and we just asked a simple question. Do they get more or less athero than the gold standard? Okay, because all roads lead to Dallas in the cholesterol world. Anyway, so I know this is real busy, but if you'll just bear with me, we kept the phenotype of elevated HDL cholesterol, whether it was on the chow or on the diet. There was definitely an elevation of VLDL cholesterol, And there was also a huge increase in triglyceride. That is a whole nother talk because I don't know anything. I'm having to learn about all this. And fortunately, I have Ira Goldberg, who is tremendously brilliant in triglyceride metabolism, to help me. But in any case, we were just focusing on this. And surprisingly, the LDLs weren't very different. Now, we don't know about size or composition, but what we did see was shown here. So these are the aortic roots of LDL, PCP2 double, and then one of our favorite animal models, which is an LDL ApoA-I double knockout. So if you can imagine, this mouse has all this LDL and no HDL. So the perfect storm of atherosclerosis. But in any case, these are the leaflets at the aortic root. So that's why we section at that region, because it's anatomically distinct. And then we stain with oil red O. And all that yucky stuff there, that is neutral lipid. And remember all those cartoons I was showing you? Well, that's the real McCoy, except in a mouse, okay? So then you quantify that, and what you notice is that the LDL receptor PCP2 double knockouts have way more deposition of cholesterol than LDL receptor, and surprisingly, as much as animals that have no HDL. Okay, so now, how does that, somebody please help me. How does that work? I thought HDL was protective. Is anybody seeing the paradox further? These animals have more HDL, right? See? Look at that. It's twice. And it's bigger, but it's not protecting.
Are there particle kinetics on any of these?
We got to make her be quiet. I'm not going to get inside your heads. Come on, ask me some questions. Come on, anything. How old I am. Did we? Thank you. Thank you. We're going to have, thank you. It's coming. Yes. Very good. Thank you. And that's what did you review that paper? No. That's what we were asked to do. Anybody? So do you guys see the problem? Honestly, I want you to see this. This is so crazy. There's more HDL, but it's not working. So in the field, we call that dysfunctional HDL. Okay? And this is like the thing now. All right, so they have neutral, I said that, and I said that too. But basically, they lack all HDL, and yet and I didn't go into the LDL, but don't worry about that. These mice actually have half the LDL as these mice, so they're really messed up. But the bottom line is, since the extent of athero is similar between mice lacking HDL and mice that have HDL but lack PCP2, then we hypothesize that HDL in the double nulls is dysfunctional. Okay, it just can't work. And you asked the million-dollar question. Thank you. And I'm sorry these are still cut off. We also looked at immunofluorescence staining. This is a CD68, and then the blue is DAPI for nuclei. And you can see the lipid staining is tracking with the macrophage staining. And that is just what people do in our field. And then because this is called pro-collagen endopeptidase enhancer protein, which is what I was sparing you, we had to look at collagen. And as you would expect, in the double knockouts, the PCP2 LDL receptor, I don't know if you can see the color, But at these, where the leaflets are anchored, you can see that there is some dark blue, which is, this is a Mason's trichrome. But notice how in the normal or control mice, there's connective tissue around. And so when you quantify that, so clearly there's not as much tissue repair and maybe not even as much collagen. So you kind of have whatever is going on with the HDL, but you also have very little repair, okay? And then we went one step further, and this is Picrocereus red, and we looked at polarizing. And this just tells you about how complex the collagen fibers are. And this is again at that leaflet that's, we did a 20x at that leaflet. I think that's right there. And I'm not sure, I guess that's that one right there. But anyway, you can see that in the PCP2 knockout, there's less, collagen gets really modified extracellularly as it gets laid down. And so these colors represent the complexity when you put them under polarizing filters. Okay, so we were at a point here. Why is it dysfunctional? You know, is it related to the collagen processing or is it something else? You know, I don't know anything about HDL and collagen, you know, putting those two together. So we had to ask, well, what Rose said, we got to look at kinetics. I mean, is the HDL particle bigger because, remember the first thing I said when we started chatting? I said it's a snapshot. HDL concentration is a snapshot. So this snapshot is saying this particle is really big. So the question is it made big or is it big because it's just like over, because it's circulating too long and it's just kind of accumulating stuff, okay? So we had to answer that. So we did a turnover study. And I know this looks daunting. I'm just going to give you the bottom line because I'm afraid I may be going over and I don't want that. I'm going to leave lots of time for questions. Anyway, what the bottom line is, so if you label HDL from the LDL receptor, or you label HDL from the double knockout, and these are distinct populations of labeled HDL, right? So we isolate the HDL from those animals, and then what you do, you put the tag on the ApoA-I, and then you re-inject those through tail vein injections, and you do that in LDL receptor or the double, and then you do the crossover, so the double knockout HDL into the single or the double. And the bottom line is these two top lines that are clearing normally, okay? I'm sorry, which are clearing more slowly. These two top lines are both these animals. And the normal die away or the normal clearance are the animals the HDL, either HDL that was injected into LDL receptor knockouts. Now, what that told us, okay, was one, there was delayed clearance. So normally when you inject these particles, right, as more fresh, unlabeled particles are synthesized, they're going to replace the radioactive ones as these are excreted. So that's why we call these die away or clearance curves. So this is time, you inject at T0, and then you look at the clearance in plasma. In these mice, it was delayed. So what it indicated is that the HDL was not really the culprit, right? Because in that case, it would have been independent of the mouse. It would have been totally dependent on the particle. But that isn't what we saw. What we saw was that it was dependent on the animal that got the HDL. So that kind of blew me away because I was, you know, HDL, dysfunctional HDL, that's where our field is right now. Everything is about the particle. And so I had to readjust my thinking. So then we looked at the macrophage and we did what I'd like to call the in vivo rater assay. Okay, so now why did I do that? Well, let me finish this thought just for you guys. So it wasn't really about the particle. It was more about the animal. So that meant I had to think more about the extracellular matrix, okay? Because it's saying that the double knockouts are lacking this protein, and this protein is involved in the clearance, okay? I mean, it was just unescapable. And trust me, I did not just jump on that. I really did not want to give up the particle. But the reviewer of the paper asked us to do this experiment. And what this is, so I told you about the Rader method where they assay human plasma for clinical trial data. Well, this is a little different. Again, you use tritiated cholesterol-loaded cells. But what you do is you inject those cells into the intraperitoneal cavity of mice. And then you look at the appearance of the tritium in their blood. Okay, now if you think about this, this is what Dan and George's Rothblatt's vision of what reverse cholesterol transport is, or efflux capacity. Think about it. Remember the plaque I showed you with all the immune cells, right? And so the idea is, what does HDL do? HDL is somehow getting that cholesterol out of the plaque and to the liver where it can be excreted. So in all their brilliance, they devised this where you're loading these cells artificially, sure, they're J774s, they're a mouse macrophage, but you load them with tritiated cholesterol. And so what you're trying to measure is the kinetics of that tritiated cholesterol getting out of the cells and basically into feces. Now, along the way, you probably go through the plasma, and that's a best bet. And so what you see here, much to my surprise, is that in the double knockouts, there's this rapid increase and almost a flattening of the amount of tritium in the plasma, whereas in the single knockout, it goes up, and then it looks like maybe it's coming down. When you look at the feces, the neutral sterols, the percent of tritiated cholesterol was greatly reduced in the double knockout. So this kind of now is starting to complete the picture, right? The HDL is not being cleared, and it's certainly not giving up its cholesterol to the liver, right? Because if it was giving up its cholesterol, one, it probably wouldn't be so big, and two, there would be as much tritium in the feces as the control. And I'm going to skip that. So what occurred to us, and it was actually a postdoc and a student working on this in my lab, so thank you, young people, and all your literature reading, was, hey, Mary, you remember SRB1? Has anybody ever heard of SRB1 besides the people who have heard of SRB? No. Any of the young people? No. Okay, so I'm going to just briefly.
People in my lab.
Yeah, they better raise their hand. Just very briefly, this was a very big deal. Gosh, I guess it was the 90s, mid-90s. And then it kind of got eclipsed by ABCA1 and ABCG1. And so Mary wasn't really thinking about it. But I know this is a lot of data, and I'm not really going to go through all this. Just listen to me. I'm sorry. I like to go to the primary data, and it looks like it's cut off. Basically, SRB1 is a receptor for HDL. Okay, just like the LDL receptor is a receptor for LDL, and it takes it out of circulation. Well, really, all you need to know about SRB1 is it's a receptor for HDL. And for many, many years, people dismissed this as just some rodent artifact. But now, finally, people have, and in fact, Dan Rader, they have finding humans that have defects in the structure of SRB1, and these people have high HDL. So think about it. If you knock out the receptor, it's not going to be removing the HDL, is it? So the HDL is going to go up, right? Just like in LDL receptor knockouts. You take the receptor out, the LDL builds up. Okay? So basically, that's all you need to know about SRB1. Here's some basic papers, 97. They have increased plasma HDL cholesterol and decreased particle catabolism. Oh my God, that sounds familiar. They have higher HDL and greater lipid deposition. Oh, that sounds familiar. So now all I had to do was connect SRB1 to PCPE2, right? Oh no. So they started doing Westerns. And so I probably should have a quiz at this point and say, okay, what do you think the SRB1 level should be? Well, we thought they'd be lower, right? Because if the receptor was lower, then the particles would accumulate. But guess what? There was more. And we repeated this and repeated this. And this is liver, by the way. So, and I was just wanting to get this paper published and get my grant funded. So then we had to start thinking about the extracellular matrix again. So how is PCP2 involved in SRB1 function? And does it have something to do with like receptor function? Okay, because obviously, I mean, if it had nothing to do with it, it would probably be the same level as control, but why is it higher? So this is it. After I get through this, we're pretty much done, so bear with me. One, I don't know that we know the answer to all of this, but this is the experiment we did to finish the paper. And so I was at a kind of a point where what I wanted to do was test SRB1 function in hepatocytes, right? Doesn't that make kind of sense? So even though the levels were twice as high in the liver, my hypothesis would have been, oh, well, they're not functioning because PCP2 isn't there. But how do you get primary hepatocytes? How do you isolate them? Anybody ever do that? I've done it before.
You digest them with collagen, so it kind of chews off all the stuff.
Thank you, Rose. So that experiment was out the door, okay? It was bad. And when it hit me, I was like happy that I thought of it, but sad because I knew I couldn't use what everybody was going to tell me I needed to do. So it was one of those mixed feelings. So what we did, and I'm sorry it's so convoluted, but I always have to tell people why I didn't do the more obvious experiment. We took CHO cells and we transfected them. So instead of, and our knockdowns were not working, so we did an overexpression. So what this is just FACS, where we stained for PCP2 using a FITSI stain. And what you see is in the transfected cells, and that's shown here with the solid line and gray, that we have more PCP2 expression. If you look at a Western, just ignore that for right now. Here is the endogenous. So CHO cells make PCPE2. When we transfected with the plasmid, which had a flag tag on it, it's larger. So that it shows that, yes, indeed, we have more PCP2 in these cells. And of course, the fact shows that it's surface, right? Because that was also an issue. It's like, where is it? It's on the surface. This is just a surface stain. So then what we did was we did that SRB1 function test. And the function test is, I'm not going to go into a lot of detail, but what it is you look at the ability of SRB1 to take the cholesterol ester from HDL out of the particle and internalize it. Okay, not just bind, but internalize it. Okay, and that's what SRB1 does. So what we saw, I think you can see, is that when you overexpress PCPE2, you get more. This is the amount of tritiated. This is a non-hydrolyzable form of cholesterol ester, so we don't have to worry about free cholesterol exchanging. So it's cholesterol oleal ether uptake. And so despite the kind of particle that we loaded, we had an increase. And then just finally, out of curiosity, we looked at the surface SRB1, and I don't really know what to make of this, but it is what we got. When you overexpress the PCP2 in these CHO cells, the profile of the antibody staining kind of shifts. So that kind of suggests, and that's where we are now, looking at the conformation of SRB1. So just to finalize this, SRB1 is a scavenger receptor. There are a large number of scavenger receptors. It's of the class B, and they form this kind of hairpin structure where they have two transmembrane domains. And it, we believe what happens is mature plasma HDL binds to PCP2, and I'm not going to go into the structure, but the grant goes into a lot of structural points. And then it somehow is delivering the particle for cholesterol ester, those are cholesterol esters, extraction, then releasing the lipid poor ApoA-I. And so go figure, right? Who would have put pro-collagen processing with HDL metabolism? And this is just some of the same stuff. It's the dysfunctionality is really at the membrane, and we believe it is required for atheroprotection and functionality. And with that, I just want to thank Omar Franconi, who was at Pfizer and now at Shire. This was his project that I heard, and we were going to collaborate, and then he left Pfizer, and I took it over. So I owe him the graciously kind of getting me up to speed. My husband, Mike Thomas, who, although I didn't talk a lot about the mass spectrometry we did with this, he and Brian Fulp help a lot with that. But this is really the work of Rakita Pollard, who just graduated from my lab, and Chris Blesso, who's now got a faculty position at the University of Connecticut. So with that, questions?
Right. I mean, do you see, is this similar to, say, like antigen presentation?
You know, so that's a great question. And the reason that I think, you know, I had forgotten about SRB1, but when you think about SRB1, what I think a lot of people forget is that the core is really extracted.
Yeah, I mean, it's.
And an LDL receptor is not like that. It is.
Yeah.
And so we really, and so of course everybody used, or I used to think, oh, well, the cholesterol ester's hydrolyzed at the surface. Well, it's not. I just told you I used an ether. So this is not like, I mean, cholesterol ester is a nasty hydrophobic molecule and is not going to sit in a plasma membrane. You know, it's incompatible with that. So how is it getting trafficked into the cell? So now one reason probably in my mind that I moved to Medical College of Wisconsin is Daisy Sahu is a structural biologist and studies SRB1 structure. And her concept is that SRB1 forms oligomers and actually works as a pore. And so how PCPE2 fits into that, I don't know. But I think I'm kind of really hoping that this is going to be, you know, a little more than just an SRB1 functional thing. So we're kind of looking at other receptors and trying to see just how global this is. But yeah, I mean, that's a good example, antigen presentation, because you have to line things up. And it's a very fast process too. Any other? Yes, sir.
Have you got a chance to look at the inflammatory status of the double knockout antibody?
You know, that is a wonderful question. And the other half of my life, I try to study inflammation. And I just, with the move and my son going to college and all these life changes, I'm a little behind on publishing some stuff, but I think there's definitely something going on there. PCP2 is made in macrophages, and SRB1 is really important in macrophage uptake of, and that's where, you see, SRB1, it not only takes up HDL, but it takes up LDL too. I mean, that's the dirty little secret. So we both knew, Rose and I both knew Dave Williams very well, and he has some publications. Of course, Dave's passed many years now, and nobody likes to read, you know, these old articles, but the truth is SRB1 takes up LDL. So it can also take up particles, but, and George Rothblatt showed this, it can also efflux. So SRB1's kind of like, you know, this bi receptor. It can take things up, but it can get rid of, so it can be both good and bad. So, I mean, that's fascinating, and we haven't quite gotten to the point where we're testing it, but we're trying to merge the projects. And we've done a lot of inflammation in the LDL receptor ApoA-I double knockout. So we're trying to put all that together. But I do know it's expressed in macrophages, so obviously, and when you hit them with like LPS or whatever, the PCP2 goes way, way up. So it must have something to do with just the architecture of the cell in an activated form, kind of like stuff that I'm going to talk to other folks about today.
So does it pay to have HDL measured?
In our field, it's kind of like, no. I mean, you know, I can't speak for everybody I work with, but it's really not. I mean, people really don't think it tells you much. I mean, obviously it's telling us something. Look at the beautiful data. And, you know, you do these clinical trials and it comes up every time. So statistically, it's saying, yes, there's an association. But whether you can use it as a ruler and say, okay, you have, you know, a 50 or a 60, you're okay, HDL. I think that's where it all breaks down. And I really don't know, you know, why. I mean, we were talking about statistics this morning. I think it's a great tool, but at some point, you know, we need better stuff, because this is really hampering drug development. Because, you know, the companies want to develop drugs, but they have to have benchmarks to show their drugs working. And so the way to image plaques is very cumbersome. And I was telling folks, you know, the heart's beating, and you're looking at these tiny millimeter wide vessels. It's a nightmare. And who wants to have angiography? It's very painful and dangerous. And it's not really all that accurate, actually. So then IVUS was developed at Cleveland Clinic, and that only looks at what's protruding into the wall. So you know, it's really tough going. So that's where I was telling Rose, most of the people I know in my field, they've started companies to different ways of measuring functionality of HDL. Yes.
One last question.
Hi. Yeah. So, you know, I wish it was an enzyme, but it's just an enhancer protein. And I don't mean that in a negative way, but so this netrin-like domain binds to the heparan sulfate proteoglycans. That's been shown in the literature. The working end are these cub domains. And those are, if you're a structural biologist, the cub domains are all over the place in nature. And they're coordinated by a calcium. And I think of it kind of like a hand or tweezers. And I think the working end of this is protein-protein interaction. And you just got to get everything in the right area. And in a sense, it's that also for HDL production, but I think that's the case for HDL catabolism. So in terms of activity, I think it's making sure that PCPE2 is on the surface, bound to heparan sulfate proteoglycan, and that the HDL gets into proximity. Did I answer your question?
The PCP2 knockout mice is function like the CETP inhibitors.
ATP, CETP, yes, yeah, yeah, that's right. Oh, I know. And that's why when all the drug company people would come talk to me, and they do, they don't really listen, but they come talk to me. I said, I would not inhibit CETP because you're backing up the system. Now, mice don't have CETP, okay? Humans do. A lot of other models do. But yeah, I mean, to make an HDL bigger and to back up its concentration by limiting catabolism, to me is never an answer. I was always on the synthetic end. My dream was to raise A1 levels synthetically and just pump out more A1. And that's a whole nother story that we have, and it does work. But drug companies don't make money off of producing, they inhibit. That's what they do best. I don't know.
Can I ask, are you planning to make some mice that maybe lack what heparan sulfate or start knocking out other parts of this
Yeah.
extracellular matrix?
I
and
do,
seeing-
yes. Absolutely, great idea. And thankfully we got funded to do some of that. So we're working on that right now.
Do you think anything like LRP, I mean, that has
Oh, yes.
subdomains all over it?
Yes. Yeah, and I was even thinking about Steve's protein, and so Ira's going to help us with the lipase. And we have some triglyceride data that is not published, but it looks like that, and I don't know that much about it, but lipoprotein lipase, maybe even hepatic lipase, but some of the lipases bind to heparan sulfate and other important proteins, and clearly PCP2 must be involved because Ira, on a first pass experiment in some mice, we had reduced LPL activity. Yes. So that's, he thinks, and the VLDL, he says it's a remnant clearance defect, blah, blah, blah. So anyway, so we're thick lately because, and we're doing a study right now and getting ready to send him like 20 different mouse tissues and all this stuff. So yeah, so I'm having to learn more about all this than I ever thought I would.
Let's adjourn now, thank
Thank
you.
you.
Students will have lunch with her back in Peter and Jill. And thank you all for coming. You sign the sheet and see you next visit. Thank you very much.